Yeah, so I'm just curious. I'm kind of looking through the chat. I don't see any major questions, but what type of queen rear methods do you guys use? If you guys have done any. Yeah, maybe there's a version similar to the Miller method that's more for small scale beekeepers. Or maybe some of you used the swarm box before. Have you tried it yet, Tisa? Yeah, VK, really try it. Yeah, I tried different methods. I really like the cloak board. I think it's pretty practical for a lot of beekeepers. And the only thing you need to do is buy the cloak board, and it just allows you to, if you have any failure, to combine those colonies back into the same colony, or make your split, or turn into a cell builder, or a queen bank, for example. So you have a lot of opportunities. As Autumn Paul says, I like to cut out swarm cells. Yeah, that works too, right? So if your colony is producing swarm cells, you can make a swarm split, or you can move those swarm cells over. Yeah, so Ski does the exact same thing. So that definitely works. It just depends on how many queens you plan on raising, or if you just want to have fun and try it out. I really love grafting. Grafting is really fun. I really enjoy it. So if you want to give it a shot and try it out, you definitely can. Okay, so now we'll get into actual queen breeding. And so we talk about queen breeding, right? There's sort of a difference between queen rearing and queen production versus actual queen breeding. Now, they can be the same in some way, but if you want to actually have your own breeding program, which some of you may not, but I'll kind of talk about like how we're having our breeding program, and maybe eventually you'd like to have your own breeding program. I'll talk about how to do that. But specifically how to do instrumental insemination. So one thing with a breeding program that you may want to do is control the genetics of the queen, but also the drone, because one of the major issues with honeybees is you can't lock them into a pen and have them mate with each other. The queens and drones are flying out, and so it's nearly impossible to know which drones the queen's mating with. And so in some circumstances, if you have a breeding program, you might want to control which drones the queen's actually mating with. And so one way to do that is through instrumental insemination. So closed mating So close mating is extremely difficult for honeybees, and that's what's made honeybees great in a lot of ways because they have a high genetic diversity, they have this multiple mating system, and that really allows a honeybee colony to be a honeybee colony. But there's also a lot of limitations with, the mating system of honeybees, right? We can breed for a trait, but let's say you're buying a breeder queen, for example, you're buying a stock from a beekeeper. You know that after the next generation, if that colony raises a new queen, that queen will fly out and mate with potentially unrelated drones or drones that have the right genetics. And so it's really easy to lose the genetics that way. And so in some situations, you might want to do close mating, which means you're basically controlling which drones the queen's mating with. And so that allows you to maintain purity of your desired traits. So if you're basically have a breeding program and a selection program for real sense of hygienic behavior or other hygienic behaviors or grooming behavior, you might want to control the genetics so you keep those genetics because you could buy a line, but if you don't control the mating, then you could lose those genetics really quickly within the next couple generations. Better performance and enhance queen productivity and temperament. So allows you to basically select for very specific traits and get the desired traits that you're looking for as a beekeeper. So whether you're producing them for yourself or for other beekeepers, it allows you to produce bees that really fit your area. And we always talk about these locally adapted stocks, like which bees and which breeds perform better in your area. And you can buy bees from a lot of different sources, but if you don't have a beekeeper in your area producing queens in your area, they may not be adapted to your specific region of Texas. But probably the major reason is pest and disease resistance, so strengthening Varroa and deformed wing virus resistance, or even other disease resistant like traits. And so for some of these traits, you might have to do closed mating. Now, with Minnesota Hygienics are a really easy trait to select for. You can do that without closed mating. You can have open mate queens and still breed from that because a lot of the genetics are inherited by the queen, but a lot of the genetics are inherited by the male, as we talked about a couple weeks ago. So with honeybees, there's a lot of challenges with the mating behavior. So like I mentioned, there's definitely a lot of benefits for the mating behavior of honeybees. The queens mate with, quite a few males, 12 to 10, or not 12, 10, 12 to 20 at times. And so this increases genetic diversity within the colony and it allows them to be better fit for a lot of these challenges. But mating can also be a major challenge, especially if you're breeding for very specific traits. And so multiple mating basically means that queens are mating with many drones, making it hard to control because the queens are flying out to these DCAs, flying with multiple males. You can't control exactly who the males the queens mate with. Also, drone availability. And so, I always say the average is around 12 males a queen mate with, but this is just an average. And so some queens may mate with only a few males and some may mate with 50 males. And it really depends on the weather conditions and the type of queens. And so you can't control exactly how many drones the queen's mating with. You also can't control the quality of the drones the queen's mating with. So at least with a closed mating system, you can control exactly the number of drones, how much sperm, and the quality of sperm that the queen's being inseminated with. The uncontrollable flight, right? Mating occurs in the air. It's unpredictable pairings. And so you might have a really good queen, but you may be getting, I mean, let's say you're looking for aggression, for example. Like you want colonies that are less aggressive. Your queen, the one that you're grafting from, may be less aggressive, may be really docile and easy to work with, but if she mates with a male that has the defensive genetics and those aggressive genetics, that could cause the workers in that colony to be more defensive. And so one problem is you might get desirable traits in your colony because you can't control exactly who the queen's mating with. And then, one thing is the seasonal weather limits, right? Like I mentioned in the previous section, that we can't control the weather. We can't control the rain. We can't control the wind. We can't control any of this stuff. And so, if that occurs, that can impact the queen mating. It could cause her to mate with less males and cause some issues downstream. And so, one thing with close mating is you can control that. Now, one thing I would say with close mating, it's more to fix a trait in your stocks, and then you graft for those to put the genetic diversity back into your stocks. And so you can't inseminate all your queens. I mean, if you only have 5 or 10, you definitely could. But if you have a lot of colonies, you're not going to have all your colonies disseminated by queens. It's just not practical. But it's a good way to fix that. And so one way to do closed mating with honeybees, and because we don't have, I mean, especially in the United States, there's very few isolated areas. And even if you're in isolated areas, there's probably quite a few feral colonies that you're producing drones that your queen could be mating with. And so it's nearly impossible to find truly isolated areas here in the United States. Now, that's to say you can't find them. So maybe some beekeepers have isolated areas to some extent. But in a lot of cases, especially here in Texas, and because of the number of beekeepers and number of bees, it's nearly impossible to find an isolated area. And so one thing you may have to resort to is insemination. So that's one thing we're doing at our Overton facility is breeding a bee and using this closed mating system to basically breed better bees, breed bees that are resistant to Varroa, tolerant to viruses, more productive, things like that. So insemination allows you to control the purity and desired traits, allows you to enhance the health, productivity, and temperament of your colonies, but more importantly, Varroa and deformed wing virus resilience. So this is what it looks like when you inseminate a queen. This is kind of ventral valve hook. So you're opening up the queen, you're grabbing the sting, and you're basically exposing the oviduct. You're using the syringe after the semen collection, inserting to the oviduct, and so she stores that into her spermatheca. So you're basically grafting from your breeder queen, so you know the genetics of the queen, and that colony may be something you really like, and then you're choosing drones from other colonies that you like, and inseminating the queen with somewhere between 10 to 20 drones, probably like 10 to 20 microliters, which is 1 microliter per drone. So insemination was first successfully, I guess, developed and demonstrated by Dr. Lloyd Watson in 1926. You're probably wondering, like, why do we call it instrumental insemination instead of artificial insemination? Dr. Watson, he hated the term artificial. And so even though a lot of the cattle producers, for example, when they do insemination, they always call it artificial insemination. And he always hated the word artificial, so that's why he calls it instrumental insemination. But they're very interchangeable, so you can call it artificial insemination or instrumental insemination. They mean the exact same thing. And so, I mean, the only reason it's different is because this person hated the term, so he decided to call it instrumental insemination instead. But yeah, ever since then, insemination started being widely used around the 1940s, 1950s. Started the development of some of the breeding programs in the 1950s, mostly for like foraging type traits like honey production, pollen, even disease resistance, because before then we didn't have Varroa or some of the diseases that we have now. And so a lot of breeding programs before Varroa were focused on production like traits and breeding bees that were more resilient. So this is what insemination equipment looks like, and I would hope at some point we can have some insemination courses across state Texas. And so, if you're interested in just learning insemination, whether you want to just kind of play around with it and see what it's like, whether you want to use it for teaching at schools, or whether you want to do it for other things, or maybe for your own breeding program, this is what the typical setup looks like. Now this setup right here, this is known as a Schley system on a dissecting microscope. Dissecting microscope about 10 to 20 x magnification. You can basically buy these microscopes on Amazon, and you can buy the Schley equipment from maybe some from bee supply stores. They tend to be more spendy, so this whole thing will probably cost you probably a few thousand dollars at least. But the setup typically has, so here's a compound microscope, so you have way to view the queen and view the insemination process. B, this is the ventral hook. So you placing the queen in this queen container, and this ventral hook allows you to open the queen, expose the oviduct, so you can inseminate the queen. The C is the stinger hook. And so the stinger hook allows you to grab the stinger and open the queen. So you basically grabbing the stinger, pulling the queen from one direction, grabbing the ventral hook to open up the queen the other direction and exposing the oviduct. The E, this is the queen holder. So you place the queen upside down and you want her to be faced with her abdomen facing up. And then right here is CO2. So what we do is we add CO2 for a minute to 4 minutes. Um, and this basically knocks out the queen during the semination process because you don't want the queen to be moving around at all. Because if she's moving around at all, you risk on kind of tearing up her reproductive tract. You risk damaging the queen, and so you want to make sure she's knocked out. But also CO2 is really important because insemination is a very unnatural process. And so one thing that initiates that sperm uptakes when you inseminate the queen for that sperm to be stored in the spermatheca is that CO2. But also we have to go back 24 hours later and give basically knock out the queen with CO2 again so she can initiate egg laying. So CO2 is a really important process for the sperm uptake and basically the egg laying. So CO2 is a really important part of this. And then we have our syringe. So syringe is where you're basically taking up the sperm from the drones, and it has like a fine tip that allows you to go into the oviduct and push that through the oviduct into the spermatheca. So once you put it into the oviduct, the queen will suck that sperm up and store it into her spermatheca. So this is what typically the process looks like. This is what we like to call a Schley system. This is probably most common. It's really flexible and a really great system to use. So typically, most beekeepers of the insemination, they'll use this Schley system. So there's some benefits for it, but there's some downsides to it. The major benefit is it's widely used, so it's really portable, really easy to use. It has fine precision, so it's really easy for a new beekeeper to pick up and inseminate a queen. Also, a wide range of flexibility and adjustments. But there are some limitations here, is that this capillary tube and the tip are not connected to each other. So the issue for that, especially for new beekeepers, is, you know, and I've showed some videos of this too, because if you're not sucking up the semen and you're sucking up the mucus proteins, for example, that can clog the tip. And if you clog the tip, then you basically lose all that sperm. And so, in some ways, you have to be pretty precise in how you collect that semen because if you clog out the tip, that can cause a lot of issues downstream. And so Harbaugh, which he was a research scientist at the USCA Bee Lab in Baton Rouge, he developed this Harbaugh syringe. So it's still the same Schley system, right? It still has the ventral hook, still has the sting hook, and still has a syringe. But instead, the tip is detached, so separate from the actual capillary tube. The capillary tube is attached to this device that allows you to bring up more sperm. Now, the reason this is a really great, so if you're going to buy an insemination kit for some reason, whether you're breeding or whether you want to learn more about it or whether you're using it for teaching, I would highly recommend having the Harbo syringe, which is this device on this Schley system. Because what happens is you can remove this tip. So let's say you clog the tip with the mucus proteins. You can take this tip out and clean it with bleach to clear out the system, flush out the system, and use it for insemination. But more importantly, you have this separate capillary tube right here. So you can pull up the semen and store it in this capillary tube, and then you can use and store that in the capillary tube for multiple days to multiple months if possible. So if you may be extracting sperm one day, and this allows you to basically inseminate queens multiple weeks and multiple months later on. So this allows you to store sperm and move for longer periods of time. Whereas for the Schley system, with this Schley syringe, you basically have to inseminate the queen the same day. So this basically gives you more flexibility. Also for this, you can only basically take up about 50 microliters of sperm. So that only allows you to inseminate about 5 queens. Whereas this allows you to inseminate about 100 queens. So it allows you to, inseminate a lot more queens. So what I'll typically do is one day I'll basically extract the semen from a bunch of drones and fill this whole capillary tube up, store it, and then come back the next day and inseminate a bunch of queens if I want to. So the Harbo syringe just gives you more flexibility, and more handling and storage capacity than the actual Schley system. But if you're only doing a few, maybe inseminate just a few queens per day, maybe 5 or 6, the Schley could be a really great system for that. It just depends on what your goals are and how many queens you plan on inseminating. So this is what it sort of looks like, right? So you have the tip, and then you have these connectors right here, these Tungine connectors to this whole capillary tube. And so inside this capillary tubes allows you to bring the semen in here and store it for longer periods of time. And so what you can actually do is you can deconnect this tip and deconnect this tube, and so all the semen is stored in here. And you can put Vaseline on both sides to keep the sperm moist and make sure it doesn't dry out or die during storage. So it allows you to put this at on your table, for example, for multiple months and the sperm won't die. So that's what's really great about bee semen is you can put it on a table for multiple weeks at room temperature and it'll stay viable for a long period of time and it won't die. And so if you compare that to cattle, for example, or pigs, like once you get that sperm from a pig, for example, it dies within a couple hours. And so, with honeybees, we do have a lot more flexibility with insemination. So with equipment, so let's say you're interested in insemination, you definitely need the whole complete I setup. So like the Schley system, I'd recommend getting the Harbo syringe, but it kind of depends on what your goals are. So if you want to buy just a Schley system and use that, that's definitely worthwhile. A microscope, so compound microscope, somewhere between 10 to 20 x magnification. You need CO2. So some of these insemination systems have a CO2 adapter that you plug right into, but you can go to a lot of different stores and buy like welding shops, for example, sell tanks of CO2 that you can use. A saline solution is really important because you can't just use water. You'd use a special type of saline solution to make sure you don't decontaminate the sperm and cause any issue with the insemination process because one thing we see a lot of issues with new beekeepers that are trying insemination is they contaminate the queen and she dies really soon after. So making sure you're practicing sterile techniques and using a saline solution will be really important. Sterile vials is really important. Pipettes to clean out and flush out the system. And so as I get into how to clean out the whole system, we use alcohol to flush out the system and we put that saline solution in. And so we have alcohol for that. Bleach, we typically use bleach for the tips, and so that mucus, if it gets built up in the tip, especially if you're not collecting the sperm properly, that can clog out the tip. And so you may want to put that tip in bleach to clean it out. Chem wipes are basically sanitation wipes that you can use, so you want to make sure you're not using your hands or anything that can cause issues or contamination. Queen cages, and then flight cages. So, obviously, if you're taking IA course, we'll cover all this in a bit more depth, but this is just kind of a list of all the equipment you need. And really, a lot of this stuff you can get off, you can get all this off Amazon. You can get the saline solution straight off Amazon, the vials, pipettes, water, everything like that. So insemination, there's a simple saline solution. And so you can buy this, directly off Amazon. It's about 90% sodium. And so you can actually make it yourself if you wanted to. Typically, what I do, I guess, is I add antibiotics to the saline solution, just to make sure that the saline solution doesn't get contaminated and transferring anything that could cause an issue with the queen downstream. So, if I do use a simple saline solution, I'll add antibiotics to it. The problem is you can't just buy that off the shelf. You have to get a prescription to get the antibiotics, but it's really important to have that during the insemination process. But there's other formulations. So let's say you plan on storing the sperm for multiple days to multiple weeks to even multiple months. You don't want to use that simple saline solution because if you just use straight salt water, that could cause damage to your sperm and cause your sperm to die. And so in those situations, you might want to use a more complicated formulation for the storage and the mixing of the sperm. So this is a kind of a more complicated mixture, right? I mean, these are a lot of big words. I think there's some beekeepers that do mixes where you can buy it directly. Um, but yeah, I mean, you definitely want to use a more complicated mixture if you do plan storing sperm for longer periods of time. And if you do, this is something that I guess I can make and distribute to beekeepers, if you do decide to take an insemination class. So I can make a bunch and, give this stuff to you. But if you do plan on extracting sperm one day and inseminating queen later on, you definitely want a more complex saline solution. But yeah, I mean, simple one, yeah, this is typically what I do, about 0.9% NaCl, so salt water with a little bit of glucose and antibiotics, and that's more enough to, collect semen and either inseminate the same day or even a day after. But like I said, you don't want to inseminate with this solution, multiple days after because it can damage the sperm and cause some of these sperm issues. And I think, let me get a picture of it, just if you're interested in learning more about it, because I bought mine off Amazon. Yeah, so what I buy is this kind of iSaline. So the Honeywell company of Amazon, they sell it's called iSaline, and it works really well. Okay, so with insemination, contamination is really important. So it's really important to sterilize everything before your actual insemination process. So, contamination can come from anywhere. It can come from your hands, come from the air, it can come from your device, it can come as you bring queens and drones into your insemination equipment. So just realize if you're doing insemination that things can be contaminated very easily. You need to make sure everything is as clean and sanitized as possible. So I always clean off my entire station beforehand. I like to flush out my whole system with alcohol to make sure it's as clean as possible. And even for new beekeepers, I do recommend wearing nitrile gloves, because your hands are really dirty and you want to make sure you're not spreading anything to your queens during the insemination process. But I don't use gloves because it can be harder to maneuver during insemination. So I always use a little bit of hand sanitizer to remove anything that could be on my hands that could contaminate the queen. And then I always use Kim wipes, so you don't want to use this regular wipes. You want to use Kim wipes because they're desanitized. Sometimes you can add alcohol to that, Kim wipes, and let them dry out, and use those Kim wipes to dry out the tip or any part of the insemination. But just know that sterilization is really important because if you don't sterilize properly, your queens could die pretty soon after insemination. So with the whole system, right, you have the tip, you have the capillary tube, and so you want to make sure that's as clean as possible. So what I typically do is I flush out the entire system with alcohol. So I'd probably say like 90% alcohol works really well, but you want to make sure to flush out the system as much as possible and clean it out as much as possible. So I always kind of flush out one round with alcohol. Then I'll flush it out one more round with distilled water to clean out all that extra alcohol. And then I'll flush it out again with the saline solution that we use for insemination. So you're basically decontaminating it with the alcohol. You're flushing out the rest of the residual alcohol with the distilled water, and then you add in the saline solution, which will be used during the insemination process. And then for the tip, I usually wash the tip also. So I wash with alcohol, distilled water, and then the saline solution to make sure the tip's also as clean as possible because we don't make sure the tip's not contaminated for some reason. Because as you're putting it together, you're using your hands to put the whole system together, and you could risk some contamination that way. Okay, so now we get to the actual insemination part. So drones are really important, and if you ever decide to do insemination, finding high quality drones and large amounts of drones can be really difficult. So one thing I typically do is, if I have a frame that's full of drones and a kind of a honey frame, I'll put those into these drone cages and these are kind of temporary drone cages that you can collect them. So it allows you to collect the drones before you actually do the insemination process. So it gives you time to clean, collect the queens, and get everything ready. But put them in kind of these sandwich cages, and you put these into a strong colony, and the colony will take care of the drones. But you don't want to leave them in here for too long because your drones can die, and that causes issues with your drones. And so you don't want to leave them in these sandwich cages too long, especially if these jam-packed with drones. But I mean, just know that probably 30% of your drones will be mature. So if you're catching 1,000 drones, only 300 will be readily available to extract the semen from for the actual insemination process. And so you want to try to collect as many drones as possible because the worst case scenario is if you run out of drones, you need to run out to the field and collect more. So these sandwich cages work really well. You can buy these at some bee supply stores. I've made these myself, and ones I make work just as well. If you have a 3D printer, you can 3D print these also yourself. So I'll show kind of a short video of the actual semen collection from Sue Kobe. Sue Kobe is one of the best insemination in the world, and she's done a lot of great courses. I know she has a course at Texas A&M. We have a course at Juliana Rangel's lab in late May. So if you're interested, she'll be teaching that course, and she's really the expert in insemination. But let me get to the drone semen part, just kind of show you what that takes.
Size of the spermatheca and number of spermatozoa stored. Drone rearing requires equal attention and is demanding on colonies. Nutrition is important, as developing drone larvae also require a specialized, rich, high-protein diet. A plentiful supply of healthy mature drones from the desired select sources requires attention. Drones are seasonally produced, have a high rate of attrition, and are sensitive to stressors. Take care to minimize pest, diseases, and pesticide residues, as these factors reduce drone numbers as well as the viability and quality of sperm. Honeybees naturally mate seasonally and during a small window of time. This is also important for instrumental insemination. The optimal age of virgin queens for insemination is 6 to 8 days post-emergence, with a range of 5 to 12 days. Drones, which are slower to develop and mature, peak in maturity at 3 weeks post-emergence. Drones will yield semen about 2 weeks old, although this is less dense and mixes more readily with mucus, so more difficult to collect. Semen from older drones past their peak is viable, although this has been attributed to plugging the oviducts, especially when queens are confined after insemination. Follow the natural seasonal rhythms of honeybee mating habits when rearing conditions are optimal. Late season, the task becomes more challenging and quality control more difficult. As mentioned, it is optimal to establish each virgin queen in her own nucleus colony before insemination and release her directly after the procedure to allow free movement to promote sperm migration. This nursery colony holds emerging queen cells. For convenience, virgins can be aged in nursery colonies, though limit the time as much as possible. Use queenless nursery colonies with a high ratio of young nurse bees supplied with open brood and feed. Remove the cell cups of newly emerged virgin queens as some crawl back into the cells and die. As the pheromones develop and virgins approach mating age, they may be subject to injury by aggressive worker bees, resulting in chewed tarsal pads and tarsal claws, missing parts of legs, etc. For this reason, it is important to properly maintain nursery colonies and bake virgin queens for the least amount of time possible. A queen's healthy tarsal pad is pearly white and plump. Discoloration indicates injury, and this may prevent the production of footprint pheromone, which functions to inhibit the construction of queen cells. The queen's ability to hold on to the comb is also a function of the tarsal claws and pads. To have mature drones readily available, I usually collect these from several breeder yards and bake them overnight for the next day's semen collection. Well-fed cell builders and nursery colonies are a good place to hold cages of mature drones. Note that normal queen-right colonies tend not to take good care of mature caged drones. Drones drift heavily between colonies. To identify the genetic origin and age of drones, these can be confined and matured in nursery colonies or marked and allowed free flight. I prefer marking drones, as confined drones are messy at collection. Newly emerged drones are easily marked with a paint pen. Drones have a high rate of attrition and are highly susceptible to stressors. Anticipate a low recovery rate of mature drones, about 25 to 40%. Of the mature drones recovered, not all will yield usable semen. Therefore, I recommend rearing 3 to 4 times the number needed to assure a plentiful supply. In preparation for insemination, I recommend giving virgin queens a brief CO2 treatment the day before the procedure. Queens require 2 carbon dioxide treatments to stimulate egg laying. One is given during the insemination procedure, the second a day before or after the insemination. It is optimal to give a treatment prior to insemination, as this tends to initiate egg laying faster and does not interrupt the semen migration. The CO2 treatment can be given to caged virgins in nucleus colonies or to a frame of virgins in a bank. Let's get started. We'll go through the insemination procedure step by step, including some troubleshooting to help you avoid common mistakes. We will collect semen first, then inseminate the queens. The flight box provides easy access to drones. A screen top with a light above stimulates the drones, keeps them warm, and aids in final selection for color and activity. Drones must be healthy and sexually mature to obtain semen. Drones from different genetic sources can be mixed in the flight cage. Here, each presents drones collected from a different apiary of select breeders. Genetic diversity is important to maintain the integrity of a breeding program. Be sure to have a good supply of drones, at minimum, twice the number needed. During semen collection, some drones will not yield semen. Others cannot be used due to contamination. Some drones will avert when simply handled. The aversion is an explosive process, and semen can be contaminated. If the semen touches your finger or drone body parts, discard. Contamination will cause infection in the queens. The semen collection process is tedious. With practice, you will gain efficiency. It is especially important to maintain sanitary conditions, as the drones tend to defecate. Have a paper towel soaked in alcohol handy. Latex gloves are recommended, as the feces causes skin irritation. To expose semen, a version of the endophallus of the drone is a 2-step process, the partial aversion and the full aversion. This is the partial aversion. At this stage, the abdomen contracts with hemostatic pressure. A pair of horn-like structures appear, the cornua. Their orange-yellow color indicates the drone is mature. If the drone is immature, the cornua are clear, lacking color, and the abdomen remains soft. Discard and try another drone. The full aversion, the second step, exposes semen. This is obtained by applying additional pressure to the partial aversion. The semen is a marbled creamy color resting on a bed of pearly white mucus. The layers are distinguishable by color and viscosity. There is some variation with age of the drone. To gain efficiency in collection, recognize drone maturity as each is grasped. If immature, discard and try another. To attain the partial aversion, grasp the drone by the head and thorax, dorsally and ventrally. To stimulate, apply pressure to the thorax. Dependent upon the drone, the pressure required will vary from slight to crushing the head and thorax. During this stage, take care not to apply pressure to the abdomen, as this will damage the endophallus and prevent the full aversion. During partial aversion, the abdominal muscles contract, making the abdomen hard to the touch, and the cornua will appear. At this stage, notice the contraction of the abdomen and the orange-yellow color of the cornua, indicating the drone is mature. Hesitate a few seconds before completing the aversion. During the partial aversions, drones often defecate. Take care to hold the drones away from your face and away from your insemination equipment during the process. Wipe with an alcohol-soaked towel as needed. Take care not to contaminate the semen, hooks, or syringe tip. The semen is a rich medium for bacterial growth. To complete the second step of the aversion to expose semen, additional pressure is applied to the abdomen. With a partial aversion obtained, grasp the drone with your thumb and forefinger at the interior base of the abdomen. Roll your 2 fingers along the sides of the abdomen, squeezing in a strong, steady motion to the tip. The endophallus is averted and semen exposed. This pressure forces the endophallus inside out to expose the semen. It is important to apply the pressure starting at the base of the abdomen and work towards the tip. Otherwise, the endophallus can be damaged and will not evert. For efficiency and to have semen ready for collection, use one hand for each of the 2 steps. With the right hand, grab the drone and obtain the partial aversion. With the left hand, complete the eversion and position the drone under the microscope. The left hand is now free to control the syringe for semen collection. You will go through many drones. Keep them warm, with a light above and fed, with a piece of candy or honeycomb inside the cage. I wipe a little honey on top of the mesh cage for the drones to self-feed. Here's another drone. Not all drones will yield. This one is immature. The abdomen is soft and the cornua lack color. Discard and pick another drone. This drone appears mature, indicated by the color of the cornua, but yields no semen. Discard and pick another drone. A mature drone. With a full eversion and exposed semen, hold the drone downwards to prevent the endophallus from flipping back upon your finger, causing contamination. The averted endophallus can be floppy at this stage. Avoid transferring between hands after the eversion is complete. Excessive handling usually results in contamination of semen. Once the semen is exposed, this should be collected immediately to prevent drying and to minimize the chance of contamination. Prepare the syringe for semen collection. Collect an airspace in the tip to separate the saline column from the semen to be collected. Each drone yields about 1 microliter of semen. Notice the creamy marbled coffee au lait color of the semen, distinguished from the underlying layer of pearly white mucus. Also notice the semen is more fluid compared to the viscous and sticky mucus. Take care to avoid collecting mucus. This will plug the syringe tip and also plug the queen. Sometimes the semen and mucus are separated during the explosive eversion process, and sometimes the semen ends up on your finger, contaminated and unusable. This is normal and the reason to have a plentiful supply of drones. The semen must be skimmed off the mucus layer. The consistency and quantity of semen varies depending on age, nutrition, and care. The semen can appear as a dense and compact clump or as several thin, stringy threads spread over the endophallus. The semen of young drones tend to be thin and lighter in color. The semen of older drones tend to be dense and darker in color. Bring the syringe tip into focus under the microscope. If using an angle tip, check the angle is positioned downward to provide more surface area for collection. Be sure an airspace separates the semen and saline column. With the Harbo syringe, I usually collect 2 airspaces. The second is insurance the airspace is not lost, and the extra bubble of saline collects residual semen from the up and down movements. If the airspace is lost, mixing results in dilution of semen and inaccurate measurement of the semen dosage. To start collection from the first drone, proceed with about 1/2 microliter of saline in the tip. Bring the tip and semen into focus. Expel this drop of saline onto the semen to make contact. Skim the semen off the mucus layer. Move lightly over the thin surface of the semen to avoid mucus. The semen is fluid and will separate readily from the thick, sticky mucus if the tip is not placed into this underlying layer. If resistance is felt, the tip is in the mucus. Try to avoid this. Semen from numerous drones is collected. For each semen load, expel a small amount of semen from the tip to make contact and skim this into the syringe. Avoid collection of air bubbles during this process. If the tip becomes sticky or to prevent the tip from drying between drones, collect a small drop of saline, 1/2 microliter. During the collection process, avoid adding extra saline and diluting the semen. Repeat this procedure until the desired amount of semen is obtained. The column of semen should be solid, without air bubbles, and uniform in color. Light and dark areas indicate excess collection of saline, making measurement of the semen volume inaccurate. The time it takes to collect semen is largely dependent upon the quality of the drones and the skill of the inseminator. Concentrate on proper techniques with care to maintain sanitary conditions.
Okay, perfect. Yeah, this is like a full video, and so if you want to watch a whole hour video, it's pretty good watch if you're interested. But that kind of walks you through the actual like semen collection, which is probably the hardest part. And so if you ever decide to do an insemination, the easiest part is actually the insemination process. The actual semen collection is the hardest part, not only because it's harder to get enough drones at producing high amounts of sperm, but also actually getting that sperm out that mucus layer, because that mucus layer can go back into the syringe and cause any issues. Okay, so once you collect all the semen and you're ready to inseminate the queen, what you want to do is, yeah, you use this ventral hook to open up the queen, the sting hook to grab the stinger and open up the queen, basically exposing the oviduct, which is the semen. You basically put it down into the oviduct. Sometimes it's kind of like a linear fashion, so you kind of maybe go in, move in a zigzag pattern to expose to the spermatheca, and then once you're in, you can basically inseminate the queen that way. So typically we inseminate each queen with about 8 to 10 microliters, which is about 8 to 10 drones, right? But you're trying to understand nature. How many queens does a drone, how many drones does a queen actually mate with? So queens will mate with somewhere between 10 to 12 drones. So we're trying to inseminate with that many drones, but also maintain the genetic diversity. So this is what it kind of looks like under a microscope, where you have this sting hook. So you grab the stinger, this ventral valve hook. So you open it that way, and you're exposing the oviduct this way. Now it's really important to have the right angle, and so having the queen chamber and the actual syringe at the right angle will make this a lot easier. Once you have all the semen collected, and with this Harbo syringe, you can collect 1 milliliter or so. So that can allow you to inseminate about 100 queens. So once you have all the semen collected and have this at the right angle, it's pretty easy. Well, I would say easy, but some people can inseminate 40 to 60 queens per day. I've even heard people doing like 100 queens per day, so it's possible. It just takes a long time to be under a microscope for an entire day dissemination, but it's definitely possible once you have the semen collected and have that all ready. That's why I mentioned I always, for me, if I'm inseminating a lot of queens, is I'll collect a lot of semen, um, probably a day beforehand, and then next day I'll inseminate the queens. So here's some kind of insemination considerations. So semen collection, it does require mature drones, and so the drones are probably the hardest part. So like Sue mentioned, you need to have well-fed, well taken care of drones, and if you do collect them beforehand, you might want to put them to a queenless finisher colony. So the queenless workers will take care of the drones better than a queen-ready colony. You want to make sure that the queen is knocked out with CO2 twice to initiate egg laying. So she does it before she inseminates and after. So it allows you to have two of those cycles. Timing matters. So we typically inseminate queens when they're about 5 to 10 days old, right? We're kind of trying to replicate nature that best variability. So when queens go out on their mating flights, they start mating around 5 to 10 days. So this is the best for acceptance and fertility. And then sterility and contamination and sterilization is really key because you want to prevent any contamination or disease transmission directly into the queen. Now, queens can be harder to introduce, right? Insemination is an unnatural process. The pheromones differ between a inseminated versus a mated queen, so introduction can be really difficult. And one thing I tell a lot of people when they first inseminate, you may only have 20 to 30% acceptance. It takes a lot of skill, it takes a lot of time, and so it's something that requires a lot of practice. So just like grafting, insemination takes a lot of time and practice to really master. Okay, so that's kind of more the insemination part and the close mating part. But like I mentioned, we'll have a lot of more II courses. So if you are interested, we'll have some here in the near future. But I guess the question I get a lot is how do you do a breeding program? So you learn insemination, you know how to graft. Now, what's the next step, right? Maybe you identified your breeder colonies, but how do you run a breeding program? And how many colonies do you need? And how do you really get started? So, we talk about, kind of choosing breeders. And so I talked about this in the next slide. And you want to, measure the traits that you want as a beekeeper. So it kind of depends on what your goals are and what you want your bees to look like or what traits you want your bees to have. So are you interested in honey production? Are you interested in bees that are less aggressive? Are you interested in row resistance like VSH and grooming? Are you interested in colony growth, survival, or maybe some other traits? So you want to make sure you measure these traits and sometimes you might have to measure them multiple times throughout the year. So some traits, you don't want to give a snapshot. You want to measure them maybe 4 to 5 times per year, whereas other traits, you may want to measure them once. And so kind of depends on your traits you're looking for and how you want to measure those traits. So one thing that I highly recommend if you do a breeding program is one, you can measure honey production, so which ones produce the most honey, but two, do they have this real sense of hygienic behavior? So this is a sort of a form of suppressed mite reproduction that allows the bees to basically sense mites inside the cells, remove those dead bees and mites, and basically suppress the reproduction. And so by selecting for this is a really valuable way to breed for mite resistance, in within your colonies. And so if you are planning to have a breeding program, I think starting with a VSH is a really great trait to add. It can be a little time consuming, take a little bit of skill to learn how to do it. But once you do, you can really breed a bee that doesn't require miticides or other treatments because bees are selected for VSH are really great at controlling and keeping the varroa mites at bay. This is a sort of a short video done at the USDA Bee Lab to show what this VSH behavior looks like. So you see this bee senses that there's a mite inside the cell. So what they're doing is they're basically uncapping that cell. So using its mandibles, opening up that smell, you see a small hole right there. So you see, often with the VSH, you see a lot of these pinholes. And now that you can see it's starting to remove this pupa and start actually cannibalizing it. And then at some point you'll see this mite jump out right here. See this mite right here? It's forcing the mite out. It's disrupting the mite reproduction. So see the mites coming out. And so okay, I mean the bees are right? It has a mite in it. It's causing damage, so they're removing it that way. And see, the mite jumps on, they get really frantic and start grooming it a little bit. So yeah, it's a really great trait to select for. So that's one thing I'd highly recommend. So one thing that we do as breeders is we have these terms called SDIs versus MDIs. So SDIs basically means you're inseminating queen with one drone. And so that basically means that the genetics of the queen, the genetics of the drone, and you inseminate it that way. And that allows you to breed from one specific trait. So if you want to breed for VSH, you can graft from a one colony that has high VSH and then collect drones from another colony that has high VSH and cross them that way to get the single drone insemination. So what you're doing is you're basically lowering genetic diversity, getting that trait, and then from the daughters, they're open mating and increasing that genetic diversity. So it allows you to select from one single trait. Now, STIs and single drone insemination are really, I guess it's really hard to be really experienced to do any sort of STIs. And so this is more for the experienced beekeepers and people that have a lot of skill with it. And STIs also don't live as long, right? Because instead of getting sperm from multiple drones, they're getting from one drone, which is 1 microliter versus 10 microliters. But then, I'd say, if you're starting out, I'd start with like a multi-drone insemination. So inseminating queens with about 8 to 10 drones. So you have that diversity within the colony and basically choosing drones that have the traits and genetics you're looking for because you want to evaluate not only where the queen's being grafted from, but also the colonies where the drones are coming from. So for a small scale beekeeper, right? Let's say you have probably at least 10 colonies. I recommend with any breeding program, you probably want to start with at least 10. Obviously, a lot more colonies is better, but 10 could be a good marker you can go off of. If you have less than that, then there's just less colonies for you to evaluate and make your progress. But just know that the more colonies you have, the more colonies you can evaluate and the best breeding you can have. Because if you only have 10 colonies, you might not have the variation there to actually breed from. So if you have 10 colonies, let's say you may want to choose one breeder queen. So one colony you want to graft from. So probably your best performer, you want that to be the one you're grafting from. And maybe you're basing it based on mite resistance, like low mite counts, high hygienic behavior, VSH, honey production, things like that. And so you can choose your breeder queen based on that. And then you want to choose maybe 3 colonies, at least, that you're collecting drones from. And so these ones also have the traits that you're looking for. So you basically collect semen from drones from these 3 colonies. You mix them up in the capillary tube, and then you seminate your queens depending on how many you want to run. And so 10 can be a really great option to have a small scale breeding program. But like I said, there's a lot of limitations because you don't have a lot of colonies. And so typically when you start, you might want to have different diverse sources. So you might want to have stocks from one beekeeper, stocks from another breeding program, and find which ones perform the best, and then graft and select from those ones. Let's say you're a sideline, you have 50 colonies, you have a lot more opportunities. So the more colonies you have, the more you can evaluate and the more breeding potential you have because you have more variation there. So anything with breeding, you want that variation. You want some colonies to be really mite resistant and might want some that aren't. But if you're starting with breeder colonies that have already been selected and bred from, you might need less colonies. But let's say you have 50 colonies. You may want to top five the top 5 breeder queens, so ones that have all the traits you want, like mite resistance, performance, honey production, things like that. And then select drones from at least 10 colonies. So you get diverse sperm and diverse genetics from at least 10 colonies, collecting drones from 10 colonies and inseminating those 5 breeder queens. And then if you have larger, so I mean, the best way to start a breeding program is have somewhere between 100 colonies. You can definitely do with 10 or 50 colonies, but if you have 100 colonies, you have more potential, you can do more evaluation, and you can find really the best performers in your region. So the more colonies you have, the more evaluation you have, and the more variation you have, more importantly, especially when you start in the breeding program. And so you can identify the top 5 or 10 breeder queens. So I mean, five is probably more than enough. It really depends on how many you want to graft from. Because most commercial beekeepers, they're managing 10,000 colonies and raising thousands of queens. They'll probably have 30 breeder queens that they're grafting from. But if you have 100 and you're only grafting from 5, that's all you need to make. And so they pull drones from about 10 to 20 colonies, depending on the traits they have, pull the semen out, mix them, and inseminate those 5 to 10 breeder queens. So that's kind of a loose way you can start a breeding program. I mean we don't really have clear guidelines of how to do it. It kind of depends on your operation and hopefully once you start our breeding initiative across Texas, we can find ways to better support beekeepers and provide more informed decisions about how to do a breeding program based on your colonies, based on the traits, and based on what you're looking for as a beekeeper. Really important to have record keeping. So we're developing our queen breeding program, our breeding co-op. And so one thing it's really important to keep really good records of the traits, really good records of the queens, and really good records of what drones are being sent with the queen. So when we start working with beekeepers across the state, we'll give each beekeeper a user code about 4 digits long. They'll have an age code, so this is your insemination date, basically. So the year, the month, and the date, and then a specific number for your queen. So th this doesn't have to be 12 numbers long. It could be one number long, but you can do these different ways. But it's a really good way to track not only your ID, the date of the queen, but also unique ID of that queen to go back and look at your queens, look at the pedigree, and understand your breeding process. So one thing that we're working on is choosing breeders based on breeding values. So breeding values are basically it's basically a very fancy way of saying like what's the breeding potential of this colony versus other ones, right? And so we use statistics to help beekeepers choose which ones you want to graft and breed from. So what we do is we base all the beekeepers and we do the average. So the average would be 0. If it's plus 10, that's 10 points above the average. If it's minus 10, that's minus 10 points below the average. So what you're looking for as a beekeeper is a breeding value that's a plus number and with the highest plus number. So that gives you more breeding potential. Now for honey production, right, this could be 10 pound, this could be 50 pounds of honey. The average could be 30, and then the low mark could be 10 pounds of honey, right? And so breeding values and the actual trait can be hand in hand. What's great about breeding value is it takes account of these things that you can't measure as a beekeeper. So the best way to view that is, let's say you have 2 yards, for example. This 1 yard, the highest colony produced 80 pounds of honey. They range from, 55 to 90 pounds. But maybe in yard two, these colonies produced 110 pounds to maybe 145 pounds. Now, how do you know that, this colony that produced 145 pounds is the best producer? So maybe they're producing a lot more honey because they're in yard two versus yard one. So with a breeding value, you can basically distinguish that. So you can say that, okay, so maybe this colony that produced 80 pounds a yard in one was the best one because if you put this one into yard two, maybe it produced 180 pounds for example. So what breeding values does, it allows you to account for those biases and those environmental things that you can't measure as a beekeeper. So especially for traits like honey production, it's really important to use a breeding value versus just raw honey production. So this is what it looked like once we have everything developed for beekeeper. And so what you do as a beekeeper is you send us you basically measure your data, you send it back, and we give you a breeding value for each trait. So like I said, you're looking for the plus breeding values and the ones with the highest breeding values. So for example, this is a queen ID, this is one queen ID, right? It has a breeding value of a zero point 0.5 for honey production. But this one had a zero point 0.8. So if you're breeding just on honey production, this one has a higher breeding value. So this one you want to select from. But this one's negative 33. So that's below average. So this is one that you want to graft from. But what we'll do is we'll basically take all these breeding values and combine them into one score. And so what you're doing as a beekeeper is you're looking for all these traits at one time. And how do you choose which one? So if you're measuring honey production, aggression, brood viability, chalk brood, and things like how do you pick and choose? We use a weighted breeding value to make it easier for you to make a decision on. So we combine all these into one single breeding value. And so, for example, Queen 4 had all these traits that you want as a beekeeper. So it has a 0.41, whereas Queen number 3 had a negative 0.12. So basically just puts a simple number next to your colony. And so once you have this running, let's say you're evaluating 10 colonies, you'll want to graph from the one that has the highest breeding value and use drones from some of the other higher breeding values. So breeding cycles, right? It takes time. Um, it takes continuous selection. You can't stop. And so it takes a long time. It's kind of like you're running on a treadmill, right? You're kind of running in the same place. You make a little bit of progress, but if you stop, you're going to fall off the treadmill and you'll be back to ground zero. And so with the breeding, it takes consistent breeding, consistent evaluation to not only maintain that trait, but also continue to breed for that. And so this is kind of the cycle of breeding, and this is what we'll start developing for beekeepers that want to participate in our cooperative breeding program. So one thing we'll do is do some training. So train beekeepers of how to use these systems, how to use breeding values, and so on. And then work with each individual beekeeper to define your breeding objectives and your goals. So as a beekeeper, what are you looking for? Are you looking for honey production? Are you looking for row resistance, virus resistance, things like this? And so defining your specific breeding objectives. And then we'll talk to you about how to evaluate those traits. So how often you get to measure them. So honey production, for example, you can measure honey production once and get that. But if you're measuring one trait like aggression, you may want to measure aggression maybe 3 to 4 times per year because maybe you go on a bee yard and it's raining and your bees are more aggressive. Well, that doesn't mean the bees are more aggressive. It just means that it's raining, for example. So some of these you may have to measure multiple times. Then we'll help you define a queen ID, give you a label, help you record the pedigree, so the tracking the drones and queens through multiple years and generations, and then helping you choose which ones you want to graft and breed from. So it's kind of a cycle, right? We're kind of working with you to define your breeding program, how to measure your traits, how to record all the data, and more importantly, help you breed a bee that's locally adapted and fits the demands and challenges that you want as a beekeeper. Or if you're selling queens, how to have a breeding program that really helps and fits the beekeepers you're selling queens to. But breeding program, right? It's continuous selection, right? And I always like to view it as a treadmill, right? You're kind of running forward, but if you stop at any point and the treadmill is still going, you're gonna fly back and hit your head and go back to ground zero. And so you can never stop a breeding program. You have to continue to do it every single year. You have to always evaluate and continue to select for a trade. Okay, perfect. Yeah, so I know that's pretty dense with queen rearing and queen breeding, but I think it's really important because as we're seeing these high colony losses, we're seeing roe are becoming harder and harder to manage, and as beekeepers, we may want to get away from chemical management and chemical intervention. And so I think breeding is one really good way to do that. Next week, we'll talk about mentor and communication. We'll talk about public service hours and volunteer hours and how to submit that. And so we'll cover all that during the next session. And with that, are there any questions? Yeah, I can send the link. Let me
Is
get the link of Sue Kobe too, if you want it.
yeah, that'd be awesome. That was a great video. Even
The
just
whole thing's
watching the
great.
snippet of it
Yeah, she covers
absolutely
everything
makes
in an
me
hour.
realize.
Yeah, she's fantastic.
Absolutely makes me makes me realize I'm not ever going to be a queen breeder.
You don't want to try it?
I don't think I could do that.
It's really not as hard as it seems. You know, really the semen collection is the hardest part, but you have to be willing to sit under a microscope and just focus for a long period of time. And some people have a hard time doing that. Doug, you have a question?
Yes, yes, I do. Um, let's say you move a hive into a new yard and you're moving your hive maybe 20 miles. Are you going to start seeing your bees react differently when they start interacting with new? Well, let's say you requeen, or let's say you have a virgin queen and you move her to a new area before you let her breed, right? Let's say you take a swarm cell and you move it into a nuke and then you move it 20 miles away. Um, you're gonna see are you going to see the reaction of the drones that she mates with right away. Do you understand what
Oh,
I'm saying?
do you mean the traits? So like if she's made with drones in that yard 20 miles away, are those traits gonna be different than ones in the same yard?
Yes.
Yeah, for sure. I mean, so I mean, when you open mating a queen, it just depends on which drone she's mating with and the genetics of the drone. So, I mean, you could be in one yard and the queen's mating with drones that don't have the aggressive genetics, and those will be really docile and easy to work with. But if you move into another yard and they're mating with drones that have those aggressive genetics, those queens will be more aggressive. And sometimes, I guess some thought, maybe your question is also like how well does that queen mix with the sperm in her spermatheca? So are they using certain sperm from drones early in the season versus later in the season? I would say the queen does a really great job of mixing up all that sperm, but sometimes you might see some seasonal differences in some behaviors. So it's tough to know whether that's a sperm that she's using to disseminate the eggs or whether that's just the environment. Because we know that bees, for example, become more aggressive when they have less nutrients or are really stressed out. But sometimes the queen may be using certain sperm for fertilization, and that can impact the worker behavior through a season. Hopefully that answered your question.
Yeah, kind of. I was looking for, I guess, so I have a bee yard in Magnolia, and I'm in New Caney, and what I have a hive over there that is getting ready to probably start building swarm cells, and I'm wondering if I'm going to significantly change the behavior of a really good hive by taking those swarm cells before they break out and bringing those queens here
Mhm.
and adding them to a nuke, so many miles
Yeah.
away.
You might have more risk. So I'm not sure. Like, have you mated queens in that area before? And have your bees
No,
been
never. This is,
never? Okay.
like I said, I'm new to beekeeping in
Okay.
Texas. I've had bees in other places, but I've never had bees in this area.
Yeah. So I
There,
mean, if you're raising the swarm cells, for example, you could have them open mate in the two different yards and see if their behaviors are different. But it always comes down to the drones, and you can't really control the drones the queens mate with. Dodie, you say something?
yeah, your the distance between New Caney and Magnolia is just not that far either. So if you've
Yeah.
got Africanized drones in New Caney, you've got them in Magnolia too. So I don't think one yard over the other is going to be that much of a difference.
Yeah, that's about the case. Yeah. Yeah, especially if it's like 20 miles, it might not
Okay.
be that different, to be quite honest. Yeah, that's a good point.
Okay. Thank you, guys.
And I lived in Conroe, so I know where you are exactly.
How do you
There
queens?
was a question.
Well, sure. I'll answer Autumn's first. Yeah. So insemination course. Yeah. So we have it, I think, in May. I'm not sure if Julian has released a flyer on her Facebook page, but I think it might be like the week before Memorial Day, but somewhere around like late May or so. I'm not sure it's open yet. And what's the cost of it? Probably the whole set about $2,000. You buy a more expensive setup. So we bought one that's kind of a mobile set, so we can actually bring that out to a bee yard or a bee operation during insemination. So, but probably it's going to run you about a few $1,000s. But you can probably save some money in some parts. But probably, I mean, that whole Schley system is probably $2,000 alone. But you have to replace it, right? Once you have it, you can use it. Other question was, so how many sources of sperm do you use in a single queen when AI? So I mean, if I'm marking the drones, I know exactly like where the drones are coming from. I try to get it from at least 8 to 10 sources. I'm basically trying to replicate natural mating, right? And so I'm collecting from about eight to 10 sources. And one reason that we collect all the semen from drones the day of because we can get that mixing of the drones and some of with random drones. But we're trying to get at least 8 to 10 microliters. So sometimes you're getting less males. Sometimes it's really hard to control that unless you're basically extracting semen from 8 males and inseminating the same queen, but oftentimes we're extracting a lot of semen at one time.
So
But
kind
yeah,
of
yeah,
along those same lines,
mhm.
there was a question earlier about if your queen goes out and mates with an Africanized drone, are those semen going to be first in line?
I think
Do
a lot
they
of
all
people
stay
think that.
together?
Yeah, I think a lot of people think that's the case. There's one study that showed that where they inseminated a queen with sperm from Africanized drones and then sperm from Italian drones. They found that there's seasonal differences, and so during a certain time of the year, the bees became all Africanized because of that. But then there's another study that found the exact opposite. So I don't know. I think the queen does a pretty good job mixing up, but that doesn't say that maybe she does prefer certain sperm during certain times of year. But it's kind of tough to say. But I'd say that she does a pretty good job of mixing it up. So I would say it's pretty random for the most part.
Okay, good. That was a good answer. There was a question in the chat a while back. Thank
I
you.
wish I wish I had the answers to it. Yeah, if they get the question all the time. So I mean, there could be some truth to that, but yeah, I haven't seen any evidence for that. But sometimes beekeepers say like their colonies become aggressive all of a sudden. So maybe for those colonies that they're preferring the Africanized sperm during a certain time of the year, for example. So um yeah, so what antibiotics do you use? And yeah. So yeah, I had a question. So what antibiotics do you use, and is it not physiological saline, contact lens solution? Yeah, so basically contact lens solution, and then we use gentian. I typically use there's a few different types to use. I think Sue typically use penicillin, but I use gentian. I don't think it matters too much. And yep, autumn exactly. So queens are basically knocked out with CO2 when disseminated. So you're basically knocking the queens out with CO2, and then you go back 24 hours later to knock them out with CO2 once again to initiate the egg laying. So, like if you inseminate a queen, and they put her back in the colony, and you don't use CO2 to treat her again, then she won't lay eggs. And so sometimes actually, if you inseminate a queen and she's not laying eggs within another week, you have to go back and use CO2 again to help her lay eggs. Okay, perfect. I think that's all the questions. Well, perfect. Yeah, and then once you have the dissemination course open, so some of you may be interested in taking it. I'd like to go to do some dissemination course across the state. I just to buy all the equipment. So typically our courses, I don't like to have more than 6 to 10 people, but hopefully we have some more in the future. Well, perfect. Yeah. So next week we'll talk about mentoring communication, and we'll spend a lot of time on volunteer hours and public service hours. And so we'll talk about some of the changes we're making and some potential modifications we're making. So, well, thank you guys all for attending, and I'll see you guys all next Tuesday.